1 is able to show us how membranes are fluid and flexible. The first thing we do is that we lift the frame from the bubble solution. When doing this, we will be observing how the bubble will grow and eventually form part of the frame. Once we bring our frame up, we have to bounce the bubble up and down. This will show us the elasticity. Then we rotate the sides of the straws and we're able to see the light reflected into the film, which shows us how the molecules move in the cell membrane. Therefore, concept 1 was able to show us how membranes are fluid and flexible. They are not static and they bend to adapt to the changing conditions that the cell membrane undergoes. Andres put his soapy finger through the bubble frame, and just like the cell membrane can repair when there are breaks in the lipid bilayer, the frame repaired as well. Now repeat the procedure with your entire hand. Concept 2 was able to show us that the lipid bilayer can be fixed by the attraction of the phospholipids in the cell membrane. Blow out on a clean straw with the other end in the bubble solution. Then lift the tip of the straw, and this will create a bubble. The bubble will grow, and then there will be a small bubble inside the large bubble. The smaller bubble will create an air compartment, but it is separate from the big bubble. In conclusion, in concept 3, we were able to see that in an eukaryotic cell, the membrane also has a phospholipid bilayer, and they have membrane-bound organelles that create compartments within the cell. The eukaryotic cell membrane and membrane of the organelles is a phospholipid bilayer. Lay the loop of thread on the film. With a pencil, break the bubble film inside the loop. The loop of thread becomes a circle, or insert your finger through the loop. Just like the pencils, pens, or fingers, the membrane proteins are also able to drift through the lipid bilayer of the cell membrane. In concept 5, we blew the bubbles through the straw, and we were able to notice that when the tip of the straw begins to separate, a tunnel is created between the bubble and the end of the straw. This allows the air to circulate between the bubble and the straw, therefore the film does not break. Concept 5 was able to show us that gap junctions are able to form animal cells that are neighboring to one another, which makes their cytoplasm be able to connect. Therefore, the material of the two cells will be able to move quickly from one cell to another. Just like the air is able to move and circulate throughout the bubble to the end of the straw. We create a bubble by blowing into the solution with a straw. Then a bubble forms and we pass a thread through the bubble, which creates two bubbles. Concept 6 was able to show us that the thread represents the ring of protein that divides two bacterial cells through binary fission.